Hi everyone, this is Genevieve Sullivan speaking. We're going to go ahead and get started here. So this is part one of a two-part webinar series about controlling Listeria in produce operations. And this webinar series is a collaboration between Cornell University and Virginia Tech. We have two speakers for you today, Gretchen Wall, who is the coordinator for the Produce Safety Alliance, so we're really appreciative that they were able to help out as well, and Alex Belias, who is a PhD student in Martin Weidman's Food Safety Lab at Cornell University. Today, Gretchen will introduce who is covered under the Produce Safety Rule versus the Preventive Controls Rule, while Alex will discuss the Preventive Control Rules as it relates to environmental monitoring. She will also talk about the basics of Listeria in preparation for next week's webinar on Listeria in produce operations and environmental monitoring. And that webinar will have some of the data that has been gathered by both Cornell and Virginia Tech on Listeria prevalence in produce operations. Today's webinar will be recorded and will be posted on the Food Safety Lab website along with the slide sets. And it's our hope that you'll feel comfortable to bring some of this information back to your own operations and perhaps use the slide sets to share what you've learned. Finally, there will be time for questions at the end of the webinar today after both speakers have presented, so please use the chat box and send your questions to me or to all of the hosts. So just send it directly to Genevieve Sullivan, or I think there's an option to send it to the panel. And I'll read them at the end to our presenters so that they can answer them. So without further ado, I'm going to turn it over to Gretchen.
All right, thanks so much, Genevieve, for that introduction. Let me get the slides moving here. As Genevieve mentioned, this is going to be a two-part series to primarily discuss control of Listeria in produce facilities and packing houses, and you're going to be presented with a lot of really great practical science-based information. But before we get into that more technical content, we thought it might be relevant to take the opportunity to discuss FSMA and how it relates to farms, packing houses, and food facilities, because one of the unintended consequences of passing the FSMA legislation is that gray area and sort of the inconsistent regulation of seemingly identical operations that pack, hold, or handle raw agricultural commodities. So in this part of the presentation, I wanted to talk a little bit about some of these FSMA definitions. And I actually got up this morning thinking about this presentation, and I had the song from Steelers Wheel. If you are familiar with the song Stuck in the Middle with You, it's pretty catchy, it's a classic. But I kind of feel like that's where a lot of packing houses are stuck in the middle with me talking about these FSMA definitions. And as it turns out, the FSMA definitions really do impact where a packing house may fall in terms of the applicable requirements. And because of those definitions, a lot of packing houses have kind of landed in this sort of gray area. So there are two definitions of a farm, but generally when you think about a farm, it's likely to fall under the produce safety rule. When you get into the facility definition, that's more falling under the preventive controls rule. And just as a very cut and dry example of when you transition from a farm to a facility would be if you're growing a raw agricultural commodity like an apple, but then you're also cutting apples to, let's say, sell to school food service. As soon as you're changing a raw agricultural commodity into a processed food or cutting it or changing it from its original shape in any way, shape, or form, that's where you start getting into preventive controls. However, that being said, when you look at the definitions that are within FSMA, not all farms were created equal, at least in FDA's eyes. So in the regulation, if you take a look at the definitions for a farm, there are two primary definitions. There's the primary production farm, which is an operation under one management in one general but not necessarily contiguous location, devoted to the growing of crops, harvesting of crops, raising of animals, or a combination of those activities. And that's, pretty straightforward. That's a primary production farm. Where you get into secondary activities farms is situations for those of you on the call that have packing houses that might be located off the farm. So this definition was really developed to address some of those facilities that might not necessarily be on the farm directly, and the definition, I'll just provide it to you now because there are some nuances that are really important to understand going forward. So a secondary activities farm is an operation not located on a primary production farm, devoting to the harvesting, packing, and/or holding of raw agricultural commodities, provided that the primary production farm that grows, harvests, and/or raises the majority of the raw agricultural commodities harvested, packed, or held by the secondary activities farm owns or jointly owns a majority interest in the secondary activities farm. So that gets into a pretty wordy definition, but you'll notice phrases like owns or jointly owns a majority of interest in the secondary activities farm. That's really what started tripping up packing houses, recognizing that that piece, the ownership piece, became very confusing to figure out whether a packing house fell under the produce safety rule as a farm or whether they fell under the preventive controls rule as a facility. Fortunately, in August of 2016, FDA did provide some clarification on farm activities, but they didn't necessarily provide clarification in terms of the under one management or in one general physical location. Some of those questions still exist today. And you may be thinking to yourself, what actually does ownership of the farm or facility matter for food safety? And when you think of it from a practical perspective, it really shouldn't, because when you think of a primary production farm or a secondary activities farm, these type of operations, they're doing the same type of activities regardless of the ownership or location, and it really shouldn't dictate which regulation the operation falls under, and it doesn't impact necessarily their food safety practices in any way, shape, or form. So there are definitely some clarifications that are needed for those farm definitions in order for farms and facilities to figure out exactly what regulation they fall under. So as a result of that, in January 2018, FDA announced that they plan to exercise enforcement discretion, primarily to allow them some time to consider changes or other approaches to address this concern of where farms fall in terms of preventive controls or produce safety rule. This means specifically that farms that were considered a secondary activities farm, except for that ownership piece. Remember, I mentioned in the definition of a secondary activities farm, there is a component that requires majority interest or jointly owns. That relates specifically to the fact that they're not subject to the preventive controls rule until the completion of future rulemaking. So that's a big relief to some of the packing houses that thought they were secondary activities farms, except for the ownership piece, because that means that they won't fall into the preventive controls rule until FDA has a chance to revisit those definitions. One thing that the enforcement discretion will not apply to is the overall compliance dates for the produce safety rule. As you can see, the majority of businesses that would need to comply with the produce safety rule have already come under the compliance dates, so 2018, 2019. Very small businesses will be next up in January of 2020, and those business sizes are based on total produce sales. I should also mention here that there are some extended compliance dates specifically for Subpart E, the agricultural water requirements, that allow 4 additional years beyond each of these compliance dates. But I just wanted everyone on the call to be aware that the enforcement discretion that I discussed in this slide doesn't impact any other areas of the regulation aside from those farm definitions. The one thing that I do want to mention is that there is some flexibility for off-farm packing houses to either comply with CGMPs as outlined in 117 Subpart B or the applicable requirements in the produce safety rule. So that's also a relief, because I'm going to go through some of the very high-level items from the produce safety rule that could apply to a packing house, or you can choose to follow CGMPs. The other thing that I wanted you all to be aware of is that when you talk about the difference between preventive control requirements and the produce safety rule, they don't necessarily have analogs that match each other. So for example, in this series, we're going to talk a lot about product testing, more so environmental monitoring, but that's not something that's required for the produce safety rule. However, I should also mention that if produce is contaminated with a pathogen such as Listeria monocytogenes, that product would be considered adulterated. So, farms and packing houses should definitely consider implementing those practices even if they're not subject to the regulation, because if pathogens found on the produce, it could be considered adulterated. So as you can see, there's a lot of nuances to the FSMA rules. And I think in the past year or two, as guidances are released and more information comes available about the FSMA regulations, I think it's easy to really lose sight of the bigger picture, which is ultimately public health protection. And I think the thing that I want to refocus your attention on is that whether you fall under the preventive controls requirements or you fall under the produce safety rule requirements, pathogens don't discriminate. They get a bad rap, but ultimately they don't care what regulation you're subject to. So there are some things that all packing houses and all facilities can do for food safety. I'm not going to go into all of the details of the produce safety rule. There's a full day training for that. Many of you might have been to that course before. But there are several subparts within the produce safety rule that are more applicable to packing houses and facilities. And those would be things like worker qualifications, health and hygiene, agricultural water, which might be used to wash produce, and then the requirements in subpart K and subpart L, which relate to growing, harvesting, packing, and then especially equipment, buildings, tools, and sanitation. So just to go over some very highlights of the produce safety rule, what you're looking at here on this slide are some of the requirements as outlined in the produce safety rule, but are things that facilities and packing houses should consider when they're thinking about reducing risk. So one of those things would be starting in the field. So thinking about harvest containers. One of the things that we'll likely talk about later in the series is limiting the ways that soil and other contaminants such as Listeria can enter a packing house. That can be done in the field by looking at harvest containers, making sure they're clean, they're not dragging dirt in from the bottom, and that they're in good condition and functioning properly. Other things that are important are correcting and reporting problems with those equipment from a worker standpoint, and not distributing dropped covered produce. So this would be produce that, for example, an apple that grows on the tree drops on the ground. That produce could potentially become contaminated and might also be damaged when it comes into the packing house, which would represent a higher risk. The equipment, tools, and sanitation section of the produce safety rule is really where I think a lot of Listeria control comes into play. You'll see familiar things that we're going to talk about in this series about equipment and tools being of adequate design. Sanitary design has been a big focus of the industry recently, especially when it comes to produce. And there's a lot of opportunity for improvement in that area. There are some requirements in the produce safety rule that focus on adequate design, including inspection, maintenance, and cleaning when it's necessary and appropriate, and also sanitizing food contact surfaces that covered produce in the regulation. And then also maintaining and cleaning all non-food contact surfaces. So think about forklifts or other pieces of equipment in your facility that might have the opportunity to spread pathogens around. Water is also one of those big areas that is focused on, especially because in a packing house, it can be a vehicle for spreading contamination that might have been a small problem, making it into a really big one. So in the produce safety rule, there are some requirements for recirculated or batch water or flume water that, first of all, it has to be no detectable generic E. coli at the start of its use. And treatment of that water is not necessarily required, but it can be used to maintain water quality and reduce cross-contamination risks. And for that reason, that's why we highly recommend using a sanitizer in all wash water, whether it's recirculated or whether it's single pass. And then another requirement in the produce safety rule that relates to changing the water helps to minimize buildup of organic material. So that makes sanitizers work more effectively, and that can potentially prevent Listeria from becoming established or at least knock back the populations. And then packing house maintenance. There are many practical things that can be done related to packing area maintenance. On the screen, you'll see lovely Dr. Laura Strawn in her native environment, which is packing house floor drains. But things like drains, cracked hoses, torn rubber door seals, conveyor belts, brushes, those are all areas where you're going to end up focusing your environmental monitoring program on. Also condensation. It's especially from walls, ceilings, cooling equipment, pipes, anything that's hanging over packing lines or in storage areas. Those can become sources of Listeria. There are also two things that aren't on this slide that I thought of afterwards that are practices that I've seen in facilities that are easy things to correct that can minimize risk. One of those is cleaning with high pressure. See a lot of people wanting to just blast everything off with a high pressure hose or some other type of high pressure water or air. That's just one opportunity that you're going to spread pathogens from one area into a place where they haven't been before. The other thing to be aware of, especially with packing area maintenance, is being aware to changes to that environment. That could be renovations, it could be new equipment, it could be a new team member or a new contractor. Anytime that something changes in that environment, it should be cause for your team to think about how is this going to impact food safety. And finally, cold storage areas. This is one area that, you get to this point, you have a clean product ready to go out the door. And I have definitely been in cold storage areas where there have been issues, especially here in the Northeast in the summer. It can be very humid, and I've walked into coolers that were just dripping with water. Anytime you have condensation or dripping onto produce, there's an opportunity for Listeria or other pathogens to grow. And the other unique thing about Listeria is that they can grow at refrigeration temps. So even though cooling isn't required by the produce safety rule, if you are using it, make sure you do it properly. So that means making sure your thermometers are calibrated and the temperatures are appropriate for your commodities. So with that, that was just a really high-level overview of some of the requirements in the produce safety rule that you might want to be aware of. But regardless of your regulatory status, you can implement practices in any type of facility to address produce safety risks. And you should be aware that even if you're exempt from the produce safety rule or the preventive controls rule, your packing house or your farm may not be exempt from buyer requirements or other industry standards. So really the best way to be prepared is to prepare yourself with as much knowledge as you can gain. And that could be attending a PSA course, Food Safety Preventive Controls Alliance course, excuse me, a CGMP's training, or any other type of food safety course such as these webinars that can better prepare you with that information. There's also a lot of great resources available out there. So for example, those from land-grant universities, Dr. Martin Weidman's lab, Dr. Laura Strawn's lab, industry organizations like PMA or United Fresh or the Center for Produce Safety. There's a ton of information. So don't hesitate to reach out and ask questions. We're all here to help in the process. So here's my contact information for the Produce Safety Alliance. We have a lot of great materials and a lot of great resources, and you're always welcome to contact me directly should you have any questions. So with that, I think I'm going to pause this presentation and I'm going to turn it over to Alex.
Thank you, Gretchen. Okay. All right, so as Genevieve said in the beginning, I'm Alexandra Belias. I'm a PhD student in the Weidman lab at Cornell. So I wanted to start giving a very high-level overview of the Preventative Controls Rule as it relates to environmental monitoring for those facilities that do fall under this rule. So the preventative controls rule basically says that covered facilities must establish and implement a food safety system or plan that includes analysis of hazards and then also develops risk-based controls for those hazards. So the requirements of the preventative controls rule at a very high level, so you have to perform a hazard analysis, you have to develop preventative controls, and you also need a system to make sure that those preventative controls are working. So the preventative controls rule as it relates to environmental monitoring and environmental pathogens really comes down to a few sections. And there really isn't a whole lot of detail within the actual regulation addressing this, but I just want to go over very briefly the parts of it. So for these first two parts, basically you have to determine if environmental pathogens are a hazard for your food products. And for most produce commodities, they are, because they are ready to eat. And they won't go through additional processing steps after they leave your facility. So if it is a hazard that requires controlling, you have to develop preventative controls. And then you also need corrective actions under this next part if those preventative controls fail. And then this section 117.165 is really where the bulk of the environmental monitoring comes in. So verifying that your preventative controls are working. And then this last section basically just says that you have some qualified individual at your company or in your plant that is tasked with overseeing all of these activities. So I just briefly want to go into what these the section 117.165 says. So this is the actual language on the environment, or what is required with environmental monitoring in the FSMA regulation. So basically it says if an environmental pathogen is a hazard that requires control for your food product, you must use environmental monitoring to verify that your preventative controls, and for environmental pathogens, this tends to be sanitation, are being effectively controlled. And then it breaks out into the different parts that must be included within your procedures for environmental monitoring. So it must be scientifically valid. You must identify your test microorganisms. So in most cases with produce, this would be Listeria species. Identify locations where the samples will be taken. So exactly what parts of your plants do you want to sample? And then the number of sites that you're going to collect. So when you go out to collect swabs, how many are you going to take? Identify the timing and frequency. So timing refers to when during production are you going to sample. So would it be after cleaning and sanitation, or would it be 4 hours into production? And then the frequency would be how often do you go and collect samples. So is it once a week, once a month? Identify the tests conducted. So are you going to use sponges? And then the analytical methods used. So would you use BAM, for instance, or the method laid out in BAM? You have to identify what lab will be conducting your testing. So that can either be an in-house laboratory, or you can send that out to another lab. And then also you should have a plan for developing corrective actions if you do come back with a positive. So as you can see, this is pretty vague language and it leaves a lot up for interpretation. So the FDA did release this document, Control of Listeria monocytogenes in ready-to-eat foods, a guidance for industry. And you'll see this is a draft guidance, so this is still subject to change. But it basically goes into more specific recommendations, and this is not a requirement. These are just recommendations on how to control Listeria within processing plants. So it gives suggestions on what should be included in a program, as well as steps for developing corrective actions. And I'm not going to go into this document because during next week's webinar, Genevieve and Laura are going to give you a lot of great tools on how to develop your own plan. So instead, for the rest of today's lectures, I want to kind of go into why, give you some more context on why we care about Listeria in our environment anyways. Okay, so each year, the foodborne pathogen Listeria monocytogenes causes approximately 1,600 illnesses, 1,400 hospitalizations, and 250 deaths in the United States. And then this makes Listeria monocytogenes the third leading cause of death in the United States. So to give you some context on how this relates to other pathogens, so this first table gives you the number or the top 5 pathogens contributing to domestically acquired foodborne illness resulting deaths. So first is Salmonella, Toxoplasma, and then number 3 we have Listeria monocytogenes. So it's pretty close to the top there with 255 deaths. And then this chart gives you the number of foodborne illnesses. And you'll notice that Listeria isn't on this list. And if you compare the 1,600 illnesses for Listeria to the over 1 million, in some cases, illnesses are caused by some of these other pathogens, it gives you, or it tells you that Listeria monocytogenes is actually quite deadly if you do acquire an illness in some cases. So Listeria monocytogenes illness. So it can have an incubation period for up to 3 months. And this is important because I know for myself, I barely remember what I ate last week, let alone 3 months ago. So it can make it very difficult to track back to exactly what food caused the illness in an outbreak situation. Symptoms can range from mild gastroenteritis, and that's what you'll normally see in healthy adults. So nausea, vomiting, aches, and fever. And then illness can become quite severe, and we label these systemic infection as listeriosis. So in these infections, Listeria can actually cross the placental barrier and cause spontaneous abortions. And it can also cross the blood-brain barrier to cause meningitis. And when we see these infections, they typically are in the young, old, pregnant, and immunocompromised because their immune systems aren't as equipped to handle the infection. So there are quite a few examples of when Listeria monocytogenes outbreaks have been traced back to produce, which is why we really care about this here today. So here's just a few of the examples. First, I want to talk about the 2011 cantaloupe outbreak. So this outbreak consisted of 147 illnesses, 143 hospitalizations, and 33 deaths. And this was linked or thought to have been linked back to the brush beds. So they found the outbreak strain in the brush beds used to clean the cantaloupes, which then transferred it onto the fruit. And this is especially concerning because one of the outcomes of this outbreak were the owners of the facility were actually subject to legal penalties. And here's an example in a produce facility as opposed to a packing house. So in 2016, there was a packaged salad outbreak, which caused 19 illnesses and 1 death in the United States, as well as an additional 14 illnesses in Canada. And all of these packages were linked back to a single facility, which indicated it was likely, again, coming from the environment. So time and time again, we have these produce-related outbreaks. And from this graph, you can actually, it demonstrates the rise in outbreaks that we're actually seeing. So on the x-axis, it's time in years. And then these yellow bars, the height of the bars indicate the number of outbreaks in each given year. And then the blue line is the trend of incidence. So we see an increase in the number of outbreaks. And this, as we can see, isn't because we're getting worse at controlling Listeria, but rather because we're becoming better at detecting outbreaks. And this is largely due to our ability to subtype bacteria. So subtyping is a tool which allows for the sensitive differentiation of bacterial subtypes. So similar to how a police officer would use fingerprinting to identify who's responsible for committing a crime, we're able to use the bacterial DNA to determine whether or not two bacterial isolates match one another. And we can use that to determine if an isolate from your food facility matches an isolate in a food product or even from a clinical case. So the two main subtyping methods that we use in the food industry are pulsed field gel electrophoresis, or PFGE, and then whole genome sequencing, which is being used more and more often. And the CDC is still in the process of switching all of their isolates over to whole genome sequencing. So pulsed field gel electrophoresis basically breaks the bacterial DNA into a bunch of different pieces, and then it separates them out based on their size. Then you can look at these banding patterns to determine whether or not two isolates are the same. So these two isolates have the same banding pattern, so that indicates that they're likely the same. And then these two are different, indicating that they are not. Switching over to whole genome sequencing, we are able to get to a higher discriminatory power because we actually get the entire sequence of A's, T's, C's, and G's that make up our DNA, or make up the bacterial DNA. So we know for a higher certainty whether or not two isolates are actually coming from the same spot. So I had already mentioned that whole genome sequencing is being used by government right now. PFGE is still a good option for industry, especially if you do have a history of PFGE patterns from your facility, because you can have a longer history of traceback. So we don't also just care about outbreaks. Those are kind of the worst case scenario. So recalls are also really important to consider when we are talking about Listeria control. So just with recalls due to Listeria monocytogenes in produce, there have been over 50 recalls since 2016. Which is incredibly concerning because it can be incredibly costly to companies and it hurts your brand image. So it's also important to think about making sure that we're preventing Listeria from getting into our products from that point of view. And the major way that we determine if we are properly controlling Listeria is through testing. So we use Listeria species as an index to indicate when conditions exist within our facilities that could eventually or could lead to the contamination by Listeria monocytogenes. So Listeria species is a more diverse group, so you're more likely to detect problems when they do occur. That's why we use this for environmental samples. And this is really where we should be focusing most of our testing efforts and resources. In the case that you do have to do finished product testing under some case scenario, you should only stick to Listeria monocytogenes testing because you don't want to be forced into the situation where you may have to recall a product when there really isn't any pathogen there. So just so we're all on the same page, I want to go over the difference between Listeria species and Listeria monocytogenes. So Listeria is the genus which encompasses Listeria monocytogenes, and there are actually, as of 2016, a total of 17 species of Listeria. So we have our pathogenic Listeria monocytogenes, which is a human pathogen, and then there's also Listeria ivanovii, which is a pathogen, but this has only been shown to date to be pathogenic in animals, so we're not as concerned about that today. And then the remainder of the species within Listeria are non-pathogenic. And then this genus is continuing to grow, and there were actually 2 additional species identified in 2018. So some of the characteristics of Listeria. So Listeria is a facultative anaerobe. So it can exist under both aerobic and anaerobic conditions. It's a soil microbe. So say soil from the field either splashed up onto your produce, or maybe it was carried into your facility on one of the bins. That could be one way that Listeria is getting into your facility. It's generally associated with ready-to-eat foods, and that's because it's killed by pasteurization or heat. So after it, you're really concerned with it after your last heat step is applied to your product. And with produce, that normally doesn't exist. It can also grow at refrigeration temperatures, as Gretchen had mentioned earlier. So say you have Listeria contamination of a cantaloupe on the outside of the rind. You want to make cubed cantaloupes. You cut your cantaloupe into your cubes, which will basically introduce the Listeria from the outside into the inside. And then you leave it in your fridge for 2 weeks. You then get into the situation where you might be making somebody sick. It has the chance to grow. So the optimal temperature range for Listeria growth is much higher, so it prefers 86 to around 100 Fahrenheit. Additionally, it prefers wet environments, so standing water in your plant is a really great place for Listeria to survive and potentially grow. It can survive stressful conditions such as high salt, and it can even persist in processing environments for over 10 years. So I want to talk about the prevalence of Listeria monocytogenes in different environments so you get a better idea of how it might be tracked into your plant. So in natural environments, and this includes samples from soil, vegetation, water, so a lot of environments that are in close proximity to the farm, Listeria prevalence is between 1.3 and 8%. So that represents a potential route. This next study actually looked at Listeria monocytogenes prevalence in urban environments. So they took samples of sidewalks, doorknobs, ATM machines, and they found Listeria monocytogenes in 7.3% of samples. So maybe one of your employees went to the ATM before coming to work. Listeria was transferred to the hands, and if there aren't proper hand sanitizing principles, then it's possible that it'll be brought into the processing environment. Livestock is also a potential route of contamination. As you can see, the prevalences in each of these, or from the bovine farms tested, are quite high. And then it can also be prevalent in processing environments, though it is quite variable based on your cleaning and sanitation principles, as well as corrective actions after finding a Listeria positive in your environment. So we're also, or a grad student in our lab is also working on another study where she collected around 1,000 soil samples from all across the United States from natural environments and tested them for Listeria presence. So 31% of those samples were positive. So we wanted to look at the distribution of those samples across the United States. So each of these circles represent where a sample was collected, and then the gray circles are negative, whereas the red are positive. So you can see it's not quite evenly distributed. And it's even more apparent in this graph of Listeria monocytogenes, where on the eastern part of the United States, it appears to be more prevalent. Though this does mean that you should completely disregard Listeria testing if you're on the West Coast, because it isn't just coming from soil, as is tested here. It can come from a wide variety of other introduction sources. So what do these sources mean for you? So it's likely that Listeria species and monocytogenes are tracked into your facilities from soil, produce, or from employees on their boots, for instance. And that's okay, we expect that to happen. Where it becomes a problem is when your regular cleaning and sanitizing isn't getting rid of them, or you aren't finding it when it's coming into your facility, and it allows it to persist in the environment. So when you do get into into a situation where you aren't finding Listeria and you can't get rid of it, standing water in the organic material in your facility is a great environment for the outgrowth of Listeria. And then the movement of equipment and employees throughout your plant can transfer it from one area to the other. And then Listeria can get into the small nooks and crannies within your facility and continue to grow. And it's really difficult once it gets into there to clean or to effectively clean and sanitize them. So you won't be getting rid of it. And then during processing, or as, again, employees move throughout, Listeria can be freed from those harborage sites or niches, which can allow it to contaminate product. And there's this outbreak that happened in Europe that was linked back to frozen corn and potentially other vegetables, that I think is a really great example of exactly how badly this can go wrong. So from this outbreak, there were 47 illnesses and 9 deaths, and it actually lasted from 2015 to 2018. So that indicates that this strain of Listeria was living or persisting in this plant for over 3 years. Because of that, the company was forced to recall over 2 years worth of product because their environmental monitoring program wasn't able to identify exactly when that Listeria came into that plant and became a problem for them. So that's incredibly costly for that company. But it is possible to control Listeria through good manufacturing practices, good equipment design, and environmental monitoring to find the pathogen. Some of these important control strategies that should be utilized to minimize both Listeria that enters the facility and then preventing it from living within this facility include sanitary equipment design. So this is really important because if you can't clean and sanitize your equipment easily, you're more likely to develop Listeria or to develop a Listeria problem. Environmental testing. So you really, if Listeria is coming into your plant, you want to be able to find it so you can get rid of it. Then also proper SSOPs are really important. And Genevieve and Laura are going to go into more detail on exactly how to develop these plans to be effective. So some take-home messages for you. Listeria monocytogenes is a hardy organism with a high case fatality rate. Listeria monocytogenes in species are commonly found in many environments, and especially with produce makes it easy to enter into your processing environment. DNA fingerprinting and Listeria monocytogenes will have a major impact of industry, so we're better able to detect outbreaks and also to identify where it's coming from, which is really important for industry to remember. And then there's also an urgent need for improved Listeria control by industry. All right, now we can take questions.
All right, thanks so much to Gretchen and Alex. So we're going to open it up for questions, so please send those in if you haven't already. And I just want to give a reminder that this webinar was recorded, and so we'll post that to the Food Safety Lab website. And we would like you to join us for part 2 of the webinar series, which will be next week on Tuesday from 1 to 2 p.m. Eastern Time, so that's Tuesday, October 15th. So Laura Strawn from Virginia Tech and myself, Genevieve Sullivan from Cornell University, will get into some of the details on what Alex presented today on Listeria and produce operations, risk factors, and environmental monitoring programs. So be sure to join us for that as well. So I see we have a few questions popping up in the chat box. I think one of them was already answered. We had a question about what is the definition of contamination with a pathogen? And so for Listeria, I think Gretchen already answered this in the chat box, but if you have any Listeria, no matter how much of it or where it is on the product, if it's contaminated after it leaves a packing house or a produce operation in general, it is considered adulterated. So there is a zero tolerance for Listeria in food. Listeria monocytogenes, I should say. Let's see, let's go to the next question. Have antimicrobial surface coatings proven to be effective in inhibiting Listeria monocytogenes growth or spore revegetation? I can actually take this one. So there has been a lot of research on this, and yes, there is some results here and there. I'm not completely fluent in that literature, but I would say that the goal is to prevent Listeria from getting onto your product in the first place. You don't want a Band-Aid to sort of help with that. So keep that in mind. But yes, there are some things coming down the pipeline in different research. I think it's all still very new and novel. But yeah, we want to prevent it from getting on in the first place if we can. Pasteurization was mentioned as a method to kill Listeria. Are there others? I don't know if Gretchen or Alex wants to take that one.
So heat is the main one that we use in industry. There are some antimicrobials, so you can go back to the surface coatings that Genevieve just talked about. And additionally, some people use antimicrobial agents or disinfectants in their wash water. However, they don't achieve a high enough log reduction to properly control produce contamination. They just prevent cross-contamination from one piece of produce to the next.
Great. And so, yeah, I'll add on to that Listeria is pretty sensitive to heat. The problem with produce is that a lot of people don't cook their produce and don't have that kill step. But it responds to bleach too. You can kill it with bleach, but again, making sure that the bleach gets into the places where Listeria is able to survive, getting into those nooks and crannies and niches and things like that. That's where we have challenges, is when the cleaning and sanitation isn't properly applied. Next question, can Listeria develop below 41 degrees? Referring to brush bed contamination in the cantaloupe case, how can we hold the brush roller distributions accountable? I've asked them how to clean their brushes and they never have any answer. That's a great question. Brush beds are very difficult to clean. Listeria can survive below freezing temperatures and it grows at refrigeration temperatures, so that's where the challenge comes in. And brush beds, there's a lot of research being done on how to clean those or to find alternatives. I don't have a good answer for that, other than I think the places where I've seen it be most effective is places that clean them every day, that remove the wax from them and then either remove the brushes to clean them or do a sort of a clean in place. But yeah, if you've ever cut a brush in half and seen the inside, that's pretty gross. So you just want to sort of stay on top of that. I don't know if
Genevieve,
Gretchen...
I was going to add on to that, just in terms of thinking about your facility going forward. You may have a piece of equipment now that you're not necessarily able to upgrade, but if you are thinking about moving towards improving your piece of equipment, you may think about, if you're going for a brush roller, think about looking for brush rollers that actually have the bristles physically molded to the handle or to the central piece. You'll look, let's say you have a brush, you can look down inside of it and you can see there's a difference in how those bristles are attached to whatever they're being held to. And if it's not physically molded to that piece of plastic, you have the opportunity to get niches for Listeria and other things that, as Genevieve just explained, you'll never be able to clean. So there are definitely some basic things, even down to brushes that you're using on your line, that you should be looking and asking those questions of suppliers. How do I clean this? And I know you said you've asked several times. Maybe it's time to find a different supplier or a different manufacturer to talk to. But just generally think about constant improvement and how you can reduce those chances that Listeria will survive in those little tiny niches.
Yeah, that's great. Thank you. And another thing to keep in mind, so Gretchen and I are both aware that brushes are very expensive. So another thing that might help is if you're able to get any organic debris off of your product before it gets sent down the brush bed. That might be another way to kind of uh, give yourself some time before you have to buy brushes again, for example, to help keep the Listeria out of getting the brushes in the first place. So if you're, for example, an apple packing house and you have apples coming in off the orchards, if there's anything you can do to have people earlier in the line that are pulling off culls, or if you can make sure that people aren't putting apples with leaves into the crates when they're out in the orchards, things like that can also help too. Next question, can Listeria monocytogenes grow in low pH, less than 4?
Yeah, so generally the cutoff for growth is 4.4. I think there are cases where it can survive, but not grow.
So the next question is the risk to dry areas. Is there any risk in dry areas, I guess is the question.
Yeah, so in general, you're more likely to find Listeria in wet environments. However, I've been in facilities before and taken samples in hot, dry rooms, and I've found it everywhere. So it's not a general rule of thumb, but when I am going into a facility and I have to prioritize where I'm going to collect samples from, I would stick with the wet, cold areas if possible.
And this is a little bit beyond the scope of this webinar, but I think it deserves to be mentioned that in a dry facility or a low water activity food, that salmonella can also be a concern. So, the same principles of cleaning and sanitation would apply for salmonella as they do for Listeria.
Yeah, those are both great points. Is there a graphic available of the physically molded brushes mentioned? Gretchen, I don't know if you have a picture of that?
I definitely do. I will need to dig for it.
Okay.
But Brent, I think that was from Brent. If you email me, I can follow up with you on that, or anyone else that's interested. I know that we've worked with some manufacturers. We have I would call it our toy box of different brushes and things that we bring to trainings, and I know I have a couple examples of those.
Great, thank you. The next question is, and I know we only have a few minutes left, so we'll try to get through these somewhat quickly. Next question is, how can we reduce the hazards of transport of Listeria from farms to the packing house itself? We may get, this might be a good question for next week with Laura Strawn. Gretchen, I don't know if you have any comments on that.
Yeah, definitely. I mean, this could be a whole presentation in itself, but when you think of some basic good agricultural practices, I think one of the biggest things that you can do is think about the flow of how things move in and out of your facility. For example, if you have a truck coming in from the field or from other areas where it might have contaminated wheels, if you have workers walking past animal pens and then going directly into a packing area. I mean, I think one of the easiest and biggest bangs for your buck is just sitting down and watching where things move on your farm, taking note of what doors people go in and go out, and thinking about how that might impact contamination within the facility or bringing that contamination in from other areas.
Okay, awesome. The next question is what is the difference between outbreak and incidence?
Okay,
Well,
so the definition of an outbreak are 2 cases of illness linked to the same organism. So that'd be the same strain of Listeria from the same food, be probably the better example there. Incidents are just single cases of new illnesses, so they're not necessarily linked to one another. So when we're looking at a decrease in incidents, we're seeing a decrease in illnesses. So that overall indicates that we're getting better at controlling Listeria, whereas the increase in outbreaks is due to just being better at detecting them.
Yeah, and I think that was a key point there that Alex just mentioned, in that outbreaks need to be detected in order to have made that graphic that she showed earlier, whereas incidents, that's probably more of an estimate, although I'm not sure on that, but that would be my guess. Next question, are you supposed to treat Listeria like seek and destroy, or is it good that during testing you are not finding Listeria?
So it's always best, it's never to look at it as a good thing not to be finding Listeria, because there's a good chance that it is in your plant. So if you're not finding it, maybe try looking in different locations or different sampling sites first before automatically assuming that it's not there at all. If you're not finding it, maybe it's a good idea to call in some outside eyes. We've done that before, going into facilities that we've never seen before and collecting samples, because maybe they're seeing something that you haven't, to really get down to the bottom if it's truly not there at all.
I think something else I can suggest is, if you're not finding it, think about all of the changes. Think about sampling more when something changes in your facility. Like I mentioned, maybe you had some renovation work done, or maybe you changed out a piece of equipment, or maybe you have some new hires. That may be a time when your chances are higher. There's a greater likelihood that you might be able to find it. So that's another way to potentially actually find it if it is there.
Okay, great. Yeah, and we'll dig more into that next week in that webinar, so definitely tune in for that if you're still having questions. We only have a couple of minutes left, and you guys are sending in some really excellent questions, so we'll try to get through as many of these as we can up until 2 o'clock. So the next question is there a possible V, B, and C, so that's viable, not cultured, state for Listeria?
So people are looking into this, though there isn't definitive answer yet on whether or not it actually exists. So most of the time, or what we should be more concerned with here, is just the Listeria that's continuing to be isolated from your plant. So those persistent strains of Listeria. And I don't know if anyone can add anything else to that.
Yeah, I wouldn't be too concerned about that. I think I would be more concerned about, what test method are you using in your environmental monitoring? Are you using a rapid test? Are you using a full culture-based method? Because depending on your method, you might have different success in actually detecting whether or not Listeria is there. And so I think I would be more cognizant of that rather than thinking too much about VBNC, although that is being investigated, as Alex mentioned. Next question. We have clients who want us to test for Listeria species instead of Listeria monocytogenes in apple and pear grading facilities. What's your opinion on this?
So if they're not willing to adjust their opinion and they want to do Listeria species, you should always in parallel also test for monocytogenes, so you can use that as proof that there is no pathogen in this product that's still safe to eat.
So I wasn't quite sure if the question was about the environment itself or the product, but we will be going more into that next week. And we do have a couple more questions, but we are at the hour, and I want to be respectful of people's time. And so I'll try to follow up with some of you privately in the chat right after we end. But I wanted to thank you all for listening in. Again, this webinar will be recorded, and there is a second webinar next week. So I'm going to go ahead and call it there. And thank you so much for participating. Have a good day.
Thanks, Genevieve. Have a good one, everyone.
Thanks, Gretchen.
Thank you.